Welcome to the Honeybee Watch Symposia series. Hello from all over the world. I'm Steve Rogenstein, the project director for Honeybee Watch and your host today. And people probably know Arrigo by now, our science director. He is behind the scenes. He's our master of technical issues and resolving them, and also the moderator of the Q&A that will be at the end of today's program. So today we'll be hearing from 3 PhD students who are at different stages of their studies, and they come from different universities and different countries, but each of them specializes in studying free-living honeybees. And today we're going to be hearing about their areas of focus, what observations they've made so far, any current data and findings that they can share, and more, hopefully anecdotes too. And once we've heard from them individually, I'll moderate a panel discussion between the 3 of them. And then at the end of the program, Arrigo will lead the question and answer session with the audience. So just to start, I'll briefly introduce Honeybee Watch for those of you who are unfamiliar. We are an international coalition tasked with better understanding how free-living honeybees survive via natural selection with little to no human intervention. And by free living, what we mean are bees that have chosen where they want to live, whether that's in a tree cavity, in a log hive, in a hole in the ground, or in human-made structures such as building walls, chimneys, or even abandoned beehives. And our aim is to find nature-based solutions to lower mortality rates of managed colonies, to increase beekeeping's sustainability, and ultimately to protect any threatened wild populations around the world. In our 5+ years, our research partners and citizen scientists have identified more than 2,500 nest sites on 5 continents, and that number keeps rising. One project that we have been working on is coming to its conclusion. After 2 years, a task force that we put together to reassess the red list status of Apis mellifera in Europe, a collaboration with IUCN, the International Union for the Conservation of Nature. We have some game-changing recommendations that will be announced next month, so stay tuned. It's very exciting for us after all this work that we've done on it. We're also part of a consortium that applied for and was awarded a 1.1 million euro grant from Biodiversa Plus and the European Commission. And that's to extensively study free-living colonies in France, Ireland, Poland, Portugal, and Sweden. And that's a project called FreeBee, which you can learn more about at free-bee or free-bee.eu, as Arrigo just dropped into the chat. And our primary mission of forming this global coalition, one of the kind of initiatives that we embarked on 3 years ago was to start the Symposia series. And it's to connect the community, including you, to one another. So now you kind of have the basic skeleton of what Honeybee Watch is all about. And now I'm going to contextualize what the Symposia series is about, especially for our newcomers. First of all, we have something like 390 people who have signed up for today's event. So super exciting. I think that that's the best attendance we've had so far. So thank you for your support. So we launched a series 3 years ago as a means to bring together the free-living honeybee community. And it was an opportunity to link scientists and experts from around the world so that they could facilitate the exchange of expertise, data, best practices, so that you, the audience, could be introduced to their work and you could link to one another. And obviously, we're committed to exploring and discussing challenging topics that we have faced and other researchers in this community have also faced. So in the first year, we explored monitoring protocols, for instance, which resulted in an open source document that's free to download from our resources web page, which Arrigo already put in there. And there, like I said, you can get all of our other videos, but also there are articles on free-living bees, natural beekeeping, tree beekeeping. There are videos, there are journal articles, there are posters. So explore that resources page if you want to get more information about these topics. In the first year, we also discussed data sharing and data rights and how to define wild in the context of free-living colonies, which is a difficult challenge because wild can be construed in so many different ways when related to bees. In our second year, we explore bee lining, tree cavities, whether wild honeybees are wild bees too, and best practices for communicating and recruiting for citizen science projects. And then this year, we kicked off in March with an introduction to Free Bee. That's that study that I referred to before from Biodiversity Plus. In June, we talked about free-living honeybees in Africa. And then in December, to round out the year, we're going to be exploring bee-related policy issues. And if you want more information about the series or to register for December or for the past so that you get the links to the videos, you can sign up on our events page, which Rigo just dropped into the chat. So that brings us to today's program, which we're calling the latest buzz, current research from Honeybee Watch Partners. So without further ado, let's meet our first guest. Matúš Pavle, which I'm probably butchering how to pronounce his name, sorry. He came to us out of the blue. He had apparently found Honeybee Watch online a year or two after it was founded. And when he was searching for a PhD program in Slovakia, Róbert Chlebo suggested that he contact us. So he did. He just wrote us an email out of nowhere, which we are very thankful for that curiosity. So at first he was inquiring about registering for the Symposia series, and that kind of brings us full circle because here he is now as a guest. But in that initial email, he mentioned that he's been tracking free-living colonies in Slovakia since 2017 and had already found 41 nest sites. So of course we were thrilled and very curious and wanted to know more. And because it was the early stages of our work with IUCN, we asked if he'd be willing to share that data for that report, and he agreed, and he's been involved ever since. So subsequently, he enrolled in the Slovak University of Agriculture in Nitra, studying under Robert, and is now involved in Honeybee Watch, helping us to write an application for a Europe-wide grant to support the infrastructure of Honeybee Watch and the network of researchers that comprise our group. So we're really pleased to have him as one of today's guests. And with that brief intro, Matúš, if you can unmute yourself and start sharing your screen.
So, my name is Matúš Pavle. I'm from Slovak University of Agriculture in Nitra, Slovakia. I'm researching interactions between free-living and managed honeybees in PhD studies. Sturgeons survived through 162 million years of changing climates, continental drifts, volcano eruptions, and mass extinctions. They resisted all but human. Today they are the most endangered species of fish as their population decreased by 94% since 1970. Golf courses along with the regular watering are in theory the perfect paradise for many species. At the same time, they are ecological trap with questionable sustainability. The destiny of all animal species is in such an environment underlined with anthropomorphic disruptive influence. The Industrial Revolution of 19th century resulted in intense tree felling. It has reached less accessible and to that time almost untouched forest areas. Many primeval forests have disappeared and were replaced with commercial forests with characteristic composition, density, and periodical felling cycles. The boom of chemicalization of agriculture started in 1950s, and here we are now. So, please think about these facts in a connection with the history and the future, not only of the species of Apis mellifera. That was my intro, and now we are going on. Since 2017, I have been attempting to find and to document the occurrence of free-living colonies in Slovakia. In the beginning, I verified which data are worth collecting. I built a nest of informants consisting of beekeepers, arborists, foresters, conservationists, scientists, and the public. Today, I observe around 100 cavities with the present or past occupancy of unmanaged honeybees multiple times a year. I'm creating the database of samples for next genomic research. I'm also observing and recording overwintering of free-living colonies in Slovakia. Since 2018, I have been conducting an observational experiment in the mountain range of Tribech. There are 5 hive locks and 13 swarm traps situated in the forests mentioned for the swarms. Most of them are deep in the woods. Every year I am observing their occupancy. I visit them monthly to pinpoint the time of survival of each colony. I document the amount of honeycombs built, the unconsumed stores. Furthermore, I take count of the dead bodies of honeybees if present. In the last 6 seasons, the occupancy of observed localities is 40% in this observational experiment. If you're interested in this part of my research, you can see the poster at upcoming Apimondia. The survival rate of these bees in this experiment is till now 0 from 2018. Last year, I have started my PhD studies at the University of Agriculture in Nitra, Slovakia. My thesis focus is on research of interactions between managed honeybees and free-living colonies. With this pilot research in Slovakia, we want to test optimal tools and procedures for data acquisition and evaluation. The outcome of this research can be used in the future as a template for a more focused study that could answer the commonalities of the living environments and overall relationships between free-living colonies and managed honeybees at apiaries. Now, something to the methodology of my research. I choose 10+1 sites with the occurrence of free-living colonies in the west and central Slovakia. Each are at least 20 kilometers, that's 12.4 miles apart from each other. The reason was to have geography, resources, and pathogens independent sites. On each site, there's from 1 to 7 free-living colonies. 1 colony at each locality is observed. The others can be used as a backup in case of the death of the main colony. We are monitoring all of the main colonies every month of the season. You can see the circles where the free-living colonies are situated. In the center of the circle is a tree with a colony observed, and visit them monthly and watch them, how they're going. This comparative research is to every observed free-living colony per one managed beehive from apiary that is no further than 1 kilometer, that's 0.6 mile from the free-living colony. Apiaries are suited in different types of environment. There are hives in the roofs of buildings in the center of the capital, backyard hives in the cities and villages, and some apiaries on the edges and landed country on the mountains too. For example, this is one of the colony on the mountains, in the tree, and on a map is the black point, the tree, and the second black point is apiary, where is the next managed colony observed. From both colonies on each side, the free-living colony and managed hive, we collect samples of bees for research of present viruses. The tests will be done by our partners at the University of Medicine and Pharmacy in Košice, Slovakia. A few of the samples will be carried over for ongoing genetic research. I collect the samples 3 times in June and September this year. I already have, and the last collection is planned in spring next year from colonies that overwintered. You can see many bees in the plastic bag because there are too many drones there, but I need worker bees for research. I prepare the samples for virology right on the site. I'm narcotizing few caged bees with CO2. This is the process of narcotization. In the plastic bag are the bees. From every bee, I take the entire intestine, and after that, I separate the abdomen. This is how I'm doing that. I take away the sting with the venom and intestine, whole intestine from the abdomen. And after this, I take away empty abdomen. I cut abdomens from 10 bees into small pieces and store them in RNA later in the freezer at -20 degrees Celsius, that's 68 degrees Fahrenheit. After completing all samples, I'll bring them in for the virological research. At the same freezer temperature, I store them, the samples for genetic research. They are in 96% ethanol. Important part of the research will be the evaluation of the present land types in the vicinity of the site of each targeted free-living colony. We'll try to find any relationship between presence of kinds of land types, their diversity, and possible positive or negative influence on studied colonies. We'll take into consideration the presence of industry and density of managed beehives in apiaries in the area. We take all the results of geographic information systems, virological and genetic research, and we'll test them statistically to find dependencies and correlations. I'm collecting the samples in this year and in the spring of 26. We should have the results of all types of research in the fall of 26. Then we should have the statistical outcomes in the spring of 27. As I mentioned before, it is a pilot research in Slovakia. We test all the parts of research, weaknesses and strengths. We evaluate their importance and needed coverage. The result will be an information base for preparing detailed research on a larger number of honeybee colonies in locations more representatively distributed in our country. The aim of such research should be to better answer the questions about relationships between free-living colonies and managed honeybees. Let me be honest and personal at the end and say something from my own history. Sometimes in the past, I was an enthusiastic sport fisher. I feel very touched with the story about one of the biggest sturgeon fish who has lived in our country, European sturgeon, known also as beluga. You can see this fish in the beginning. This fish was abundant in the river Danube, has reached length around 6 meters, that's around 20 feet, and was valued source of caviar. The last one seen in Slovakia was in the 50s. After I noticed worsening quality of the water in the rivers of Slovakia as a fisher and found that the toxicity of fish is rising, I stopped fishing completely. I started beekeeping in hope for a better and healthier hobby. As I got deeper into the topic, I found that even though the bees have more freedom of movement than the fish in the rivers, their environment is strongly dependent from us too. Free-living colonies are the perfect natural lab object for studying selection mechanisms. There are also strong bioindicators of the environment which we are changing. Can the honeybees still stay alive without our help? Have we destroyed environment in such a way that it cannot be fixed anymore? It's all about interactions and relationships between species. Studying this can help us prepare a better future not only for Apis mellifera. So now enjoy a few more shots from my observations of free-living colonies. Most of them were shooted in parks, on cemeteries, in cities and villages, in old trees. Just few were in forests. So thanks for your attention and enjoy next presentations of my colleagues.
Thank you. What a great presentation. This is actually the first time that we've had somebody do a video presentation. So thanks for challenging us and giving us all these great visuals. It's a nice complement to what we've been doing in the past.